and more people are coming. So yeah, thank you, Emmanuel, for taking your time being our speaker today at the Monash Advanced Microscopy webinar series. And to start with, I wish in the name of the Monash Microimaging Facility, but also Monash University, to acknowledge the people of the Equalian nations on whose land Monash University is located and pay our respects to elders past, present, and emerging. So to introduce Emmanuel Reynaud, I hope I pronounced that correctly. Emmanuel Reynaud is a French biologist with a thinker's mindset. He has been involved in developing new instrumentations to explore new areas of research, ranging from nanosurgery of cells to 3D fluid bioprinting. He is the co-founder of the Light Sheet community and has been involved in technology development, for example, at ZEISS, the probably worldwide biggest microscope company, in developing the Light Sheet Z1 and 7, conferences and workshops to promote the technology since 2009. His laboratory has contributed not only to lab-based projects, but also to expeditions like the Terra Oceans 2009 to 2012 and museum exhibitions. So thank you again, Emmanuel, for taking the time and giving us some good advice, like your title of 140 light sheet microscope types, a few survival tips. We are looking forward to learn much more how to use these microscopes. So the stage is yours.
Thank you very much. It's a pleasure to be able to speak to the other side very early in the morning to make sure I have a kind of a early day. So it's going to be a good day starting very early. Thank you very much. And so I just want to warn everybody, because this talk is not going to be politically correct. It's going to be about telling you how bad it can go and why. So I've been in the thick of light sheet microscopy for far too long now. And what I'm going to talk about, it's not about how great my lab is and how fantastic my ego impact factor level is, but mainly about what is light sheet and what you should know about it, because now we have reached 152 acronyms. So it's extremely confusing. So people think that a light sheet is always the same, but light sheets are very different in thickness, in length, in the way they are generated and things. So I'm going to try to give you some tips on making sense of all those acronyms and understanding what's going to work for you and what's not going to work for you. Because the light sheet is, like any microscopy technique, is working well for some things and not for others. And there is no such thing as a perfect microscopy technique. So hang on, guys. I'm going to share my screen and we're going to go to the thick of it. Can I? Yeah, can I check? Yes. Can everybody see my?
Yes. Yes. Yeah, no, it's perfect. Yes.
So first, I just want to remind you that, yeah, as I've been in the thick of it, so we piloted to Manchax in 2009, we have been running the Light Sheet Fluorescence Microscopy workshop, then conference, and now we're also running the EMBO workshop, and this year is going to be the 4th time we're running it in the Czech Republic. We have hosted several Australians image facility specialists over the time, because we are a community. So the idea for us is, as the Light Sheet community, the Light Sheet brings a lot of problem from image processing to also, we have been welcoming everybody, and that has grown in numbers, and that has been given an opportunity to share a lot of things and also develop a lot of things. So not only the technique has evolved, but in parallel, we get a huge amount of imaging specialists, especially for the image processing. So as I said, we're running the course, and the course has been growing in numbers. We have with 60 more people who run from facility to others. And here is the last conference we did for the 10th anniversary, and you can see that the numbers of people have increased in numbers. And we don't only have people dealing, sorry, it's coming a bit too fast. We don't only have optics people and physicists, we have also biologists and image analysis people. And we had a huge need of them because we generate far too much data for what we're supposed to handle. And that's one of the main problems we're going to discuss later. To help out, we wrote a couple of things like this one, probably many of them you know, it gives you an overview of what you need to know when you die to light sheet, but it's quite old now, and we hope to get another one. And as I was mentioned by Jennifer, I've been involved in creating the fridge So the Z1, that I call the fridge because you have a door and it's cool inside, and you could have a beer opener, but size refused. Now we have a 7. Remember, this one is not the perfect one. It's the one which fits as many applications as possible. So I'm still not happy with it. And maybe when we reach the Z9 or 10, we will probably go somewhere closer to what I wanted, but I will be retired by then, and then I don't care anymore. So my main part into that big plot of light sheet microscopy was sample preparations, and we did a lot of advice on how to mount sample into what originally was the SPIM systems, which is hanging the support from above. But now things have changed tremendously because you've got so many light sheets that you can do it upside down, on the right, underwater, whatever. So it becomes a very big problem for many. But I just wanted to remind you that my origin is this, field biologist. So I tend to do things in the real thing. And it's also what we're doing because we're currently developing an underwater light sheet system. So I can bring it with me underwater when I do my kind of stuff. So we are specialists in 3D biology, which means that we do those kind of things. So we do 3D coral photogrammetry, CT scan, MRI of large structure. So for example, here are corals for collections. We have done also underwater corals using a photogrammetry technique, and we've been working on the texture systems versus the point light systems. This is, for example, a way of following colonies over time using a GoPro. So basically you go around the systems, analyze, create the data, give you the volume so you can come back the next month. And now some of our methods are used in many coral expeditions in order to measure. And that's showing you one example which has been extracted from a coral and brought back to the shore for analysis. So our expertise is mainly in 3D, and it's why light sheet was interesting for us because it was able to capture what we could not capture before. Between the confocal microscopes and the CT scan, then there's a big gap, and it's what we call mesoscopy, which is appearing now as a field. And light sheet for us was what we wanted, is this kind of closing the gap. And when I encountered that, I was really happy, and I jumped on the train. So the techniques allow us, those technology allow us to have a good understanding of 3D and how you capture 3D and how you analyze 3D and how you build 3D. So for us, it has been also very useful when we went into light sheet microscopy. So here is moving from colors to fluorescence and being able to layer the data at different density and different level over time. So now light sheet. So light sheet can also be called plane of light. Some people will call it lattice. So we'll call planar illumination. Some will call it single, or some will call selective. Some will go for 1 objective, 2 objective, 4 objective. We're hoping for 8 objective one at some point, because somebody, you know, when you start that, I have a big car, somebody will have more width than you and then more horsepower. So this is a never ending games of, you know, my microscope is better than yours. I have a better resolution, and, you know, all kind of thing. We're forgetting that most part of the users don't care about the resolution. They want the resolution they need, not the resolution you're enforcing me to take. And also people have a strange view on the resolution. Is they believe that the resolution which is written on the paper is the resolution they're going to get. But it's like a car. If you get Lewis Hamilton to drive your car, it's going to be faster than you anytime, because he's a good driver and he knows how to use the car properly. If you drive a car yourself, you're never going to get the same speed than Lewis Hamilton, because you don't know how to use it as well. So be careful when you talk about resolution of my pictures. It's not necessarily what was written on the box of a microscope that you're going to get, because that depends on your sample, depends on the way you set your microscope, and in many other aspects of it. So a few years back, colleagues of mine trying to explain to people the difference between the different microscopes. And here is an example from Julien Colombelli and Corinne Lorenzo from 2014, showing you all the different kind of microscope you could analyze with light sheets. And you can see it's messy because, you know, there's so many parameters you have to care for. And this is usually not a bad, it's a pretty bad idea. Try to get, you know, the speed, you know, that's a pyramid of frustration, trying to get everything together. And that leads to one problem. Is in light sheets, we have this. So that's only a third of the acronym for light sheet microscopy. And you can see that it's pretty messy. everybody went for a kind of an acronym. Everybody wants to prove itself. And we have 140 plus. So I've added a few acronyms if you want to use them because they are free. So ground SPIM is available, Jupyter-based LSFM if you are into astronomy, the no SPIM, which is probably a confocal, the Y SPIM. Why do you need a SPIM anyway? Because you don't have one. And the ZZ LSFM, because nobody wants to use a Z in their acronym. I don't know, that doesn't work. Coco SPIM and the B SPIM are also available. Interestingly enough, if you look at the acronym bunch, you have what I call the SPIM nebula. So people lack of imagination. So they use the first paper, which is the SPIM, the Selective Plane Illumination Microscope, and add things in front or at the back. So it's an easy, quick acronym, you know. So you have a QV SPIM, the SO SPIM. So we have a DO SPIM, the D SPIM, the da, do, and so on and so forth. And you have a DSLM cluster. So, however, you have to remember that most of the SPIM acronyms are not related to the SPIM in geometry and the way it works. So they use SPIM as an acronym in part of the acronym, but actually the microscope doesn't work like a SPIM. It's not mounted the same way. It's not organized the same. So one thing I want to remind you is about what a light sheet is. So a light sheet is a plane of light. But because you have a plane of light, you define two spaces. One is a line, because each time the plane will hit a surface, it will create a line. And it's called line scanning. And this thing has been going on for years. Your barcode scanner in every supermarket is a light sheet. The alignment systems in X-ray, when you put your hands on top and you have a cross, it's also a light sheet. So a light sheet defines two spaces, a line for scanning and a plane for scanning as well. So you can actually use a light sheet microscope in two ways, scanning the surface of an object or scanning inside the object if it's transparent enough. So it means that that's line scanning. So those are all light sheets. So each time you go to an MRI, you get two light sheets. If you bar scanner, it's a light sheet. If you do alignment DIY to make sure all your frame in the wall are correct, you use a light sheet, but you use the line to scanning. But it means in a light sheet microscope, you can actually do the same. So if you put an insect or something which is refractive, you can scan the surface of an object. And we have done very tiny moth and very tiny nematodes and plankton by using the scanning aspect of a line and not the plane. And it tells you that actually light sheet illumination is the largest type of illumination you can get in the world, because you can scan Buddha. So the largest light sheet scanner is sold by Leica, and the light sheet is about 150 meter high. One is set up to scan the cathedrals. So the best one you can get is this one, is a 4-camera, 4 light sheet system. It can scan a tree trunks at 2 tree trunks a minute and allow you to cut planks. It's a very expensive one. It's a very big one. But this has been used in industry for years and you can see that the angle between the illuminations and the camera is not 90 degrees because they're reading at 45 to read the surface very quickly. So this is a German company. It's I think it's 450,000 just for unit for scanning, and you have the cutting machines and stuff, and it's about 150 meter longs once you get all of it. So it's a very big light sheet illumination systems. So light sheet is important. It can even be used by cats. So you can buy those light sheet projector system, which is very good for teaching cats. This is courtesy of Robert Haas, a colleague, which is having a cat called Pixel, which is learning about imaging in general. So this is the typical light sheet we all heard about. You see the light sheet, perfectly thin, perfectly nice, in between two objectives, one's doing the illumination, one's doing the detection at 90-degree angle. And this is the SPIM setup. So actually you get your sample from the top and you get your light sheet like this. But you actually can do light sheet like this, light sheet like this. You can actually change, as long as the two objectives are at 90 degrees, you can have them at any angle you want, depending on where your sample are. And that's also why we have so many acronyms, is if you use a SPIM, typically your sample comes from the top. If you use a lattice light sheet, it usually flat on the cover slip and it's coming from underneath. If you use a dye SPIM, it comes from above. So each time you will have a different relationship between the light sheet position and your sample. And that's something you have to really understand, is when you talk about a light sheet microscope, you need to understand the geometry, because the geometry will drive the way you mount your sample or not. because some sample will not fit. So now a bit of history. So that's probably what you remember, all know about is in 1903, there was the ultra microscope and Sidentov and things. They wanted to look at very tiny particle. And this led to quite a few Nobel Prize, because not only Sidentov and Tishbandi got the Nobel Prize for their work, but it influenced the work of others, because at the time, ultra microscopy was the super resolution at the time, and it was not in biology at all. It was purely chemistry and physics. Nobody cares about biology at the time. However, something which is very important is the ultra microscope was sold by Zeiss to look at bacteria and all kinds of parasites. So even though a lot of people in the community tend to, oh, no, it was only chemistry, we were the one bringing it to biology. Actually, the ultra microscope until 1926 was sold to look at bacteria as a dark field illumination. And here are some pictures from plant cells, which were published in 1914. And below you can see sporozoids, which are parasites of malaria, with blood cell into an ultra microscope. So it was used as a dark field illumination because as you had only one plane of light going through the sample, the quality of the image were much better. So it was not used as a scanner, it was a single plane, and you were looking at things going through a single plane. Because when you have a light sheet systems, you can move a plane or you can leave a plane where it is. So not only you can use the line, you can lose the plane, and you can lose the plane two ways. You can keep the plane and you look at what's going through the plane, or you can just use the plane and go through the stack. So in that particular case from 1913, they were using a single plane and they got gorgeous pictures because they got a gorgeous background. So the signal to noise ratio was perfect to do drawings, and there are some examples from plants and sporozoids. So biology was used in light sheet context until 1926 and probably afterwards. And they even did movies, and some movies are available in Pathé archive, and they did a big evening at the Académie des Sciences in France on ultramicroscopy images. So here is what happened to light sheet afterwards. Nobody wanted to use the light sheet because it was useless, apart people doing particle velocimetry. So in particle velocimetry, you have a plane of light and you look at what's going through. So it's a single plane, static, and you look at the movements. And it's very good because like Cismundi, you can see every single particle going through. So that's perfect for discriminating fluid analysis, fluid dynamic. So PIV, particle velocimetry, actually developed single, double camera, three camera system, 3D system 20 years before the SPIM appeared. I'm sorry, I have a fly which just don't want to. So, and for example, recently people from Scripps have been able to use a light sheet underwater at 700-meter depth by scanning a larvacean. So basically they create a light sheet on an ROV and they move it through the jellyfish and they are able. So light sheet is also the deepest imaging technique you can use to make 3D images nowadays. And this is an example from a larvacean, which are very big floating things, and you can do that. And in my lab, we are currently developing a small light sheet system for ctenophore and following jellyfish and plastic particle. This is very good. It's called Deep Field. It's the same principle, the light sheet, but will appear on a search engine as PIV and not as light sheet. So it's also the confusion around that is different groups will use different acronyms to actually compare their systems. So here is an example where we did jellyfish at 600-meter depth. So now let's go back to the thicket. One first thing you have to remember is the way the light travels through your sample is very different in a light sheet setup. In a normal setup, you go through the sample and go back to you, so you go deeper into your sample and come back, or you go through the sample, okay? So it's a straight line. That's the way the system works between the illumination and the detection. In the light sheet, it comes from the side, okay? So what you're looking at, you're looking from underneath on how the light is going through your sample. So it's a very different ways of looking at your sample. You're going from the side and you're looking at how the light travels through, while before you look at the light travel up and down. So many artifacts we see in light sheet microscope actually appear in confocal, but because of the way you could detect it, you don't see them. And that's very important. Remember, the light goes through the sample. So you get all those gorgeous drawing, you know, the thickness of a light sheet and how it transmits. You can calculate all kinds of physics. This is pure theory. This is a lovely drawings. It looks very nice in the paper. You can calculate the resolution you want. This is not the reality. It's a pure schematic diagram. This is not what you're going to get. So the thickness of a light sheet, the ways, the field of view, how you can move them and swing them around and all kind of thing. This is purely theoretical. It's easy to draw, you know, it's physics, you apply the algorithm, that's what you get. However, when you get into a sample, the light sheet never stay the same. So in that particular case, where we compare confocal to light sheets, you have the impression that the plane of light stayed perfect across your sample all the way through and you detect everything. I'm sorry, but no. So what happened is this. So on one side, you have the theory, and on the other side, you have the reality. So you can see that the light sheet probably disappeared after 150 micron to 200 micron. And the bigger your sample is, and the less clear your sample is, and the worse it's going to get. So photons are not your friends. Photons don't do what you want. Photons don't get into a sample, find the GFP, activate it, and give you the fluorescence. It's pure random. And the thicker the sample, the worse it gets. So remember that this is what happens when the light sheet goes into your sample. And if you come from two sides, it will be the same. Things in the middle will go wrong. So what happens is you get transmission, reflection, refraction, diffraction, absorption, scattering. And if you want to know more about that, we had published a blog series in Focal Plane with Elizabeth Kugler, and these 5 episodes going through the different problems you can encounter with light sheet microscopy. And I will send them to Jennifer so she can distribute if you want. They are open source, open access, and we basically tell you the truth about where it's going to go wrong. And here is an example is what's going to happen with the light. The light comes from a side, some will be transmitted, some will be reflected. And as I explained, line scanning is what's happening. Refraction is so common. So what you think you're seeing at a certain depth is actually not there. Because as the refraction, the light sheet get up, and basically what you look at is actually 10 micron below what you're looking at, because the thing is curved. And you can observe curvations of light sheets into systems. And we have done that during the course. Diffraction is also a big one. Absorption is, in some sample, absolutely terrible. And remember that it's also light wavelength dependent. So some wavelengths will be more affected than others, depending on your sample. And as it comes from a side, that will slowly vanish. So across your field of view, things are going to disappear. Or you think they are bigger or smaller. I had to reject some papers because they were seeing things which were bigger in the center of the spheroids. They never were bigger. It's just because the diffraction and refractions increase the light sheet thickness. And what they were looking at was 5 times the size of a light sheet from a side. So what happened is that you have light interaction within your sample. So you have noise, uneven excitation, acquisition artifact, and variation in point spread function, because as the light sheet goes through, things are changing and the light sheet gets thicker, diffract, diffuse. And also remember that the photon generated by fluorescence don't come to your camera in a straight line. So the deeper you go into your sample, the same thing happens in the other direction, because the photon has to try to get out of a thicker sample toward the camera. So you have 2 problems. One of the light sheet going into your sample, and then of the illumination, the photon trying to go back to your camera. So those things are 2 things you have to consider. So we have a kind of a dual problems. One on the illumination getting deeper and diffracting and diffusing into your sample, and the same thing in the fluorescence, because we're in the fluorescence mode. So what we're looking at are excited photon going up to the camera. And the deeper they are, the more difficult it is for them to find the camera. And sometimes they don't find it. So the deeper you get, so that's the example here. As you go into a thick sample, the deeper you get along the illumination axis, the thing gets worse. And also for the light sheet. So you can see that as you go deeper, the light sheet doesn't penetrate as well, but also the detection is getting worse because it's thicker and thicker for the photons to go back there. So when you combine even with two, it's getting better. You can see photon a bit further down into your sample, but after a certain depth, what you're recording is useless. And what's going to happen to you is because, oh, there's a light, you know, the pure GFP thing. As long as you see green, you think you're right. But what's going to happen is often you're going to record the entire spheroids, but the back of the spheroid will be so diffracted, diffused, that the PSF will be 10 times the size of a PSF in front. And then after, you're going to try to fuse that, a good front with a bad back, and that's going to go very bad because the computer is going to try to make up of what is that thing? And then you're going to average out your data. So it's not because you see the entire spheroid or your entire zebrafish that it's worth recording. Just remember this, the deeper you get, the worse it gets in both directions, light sheets and detection. And that's always the case. And it's getting worse and worse because you have automated fusion of both light sheets. And when you actually project them, you realize that something at the back is not looking good. So be careful. It's not because you see the GFP until the end of the world that it's worth looking at it. No, don't follow the green light, you know, don't be, you know, like cats, you know, you use a laser pointers and they follow everywhere. It's not a good idea. Think before to record. So what you have is a combination of different things. You have excitation decay, detection decay, both of them are, and you have to make a kind of a equation between the two. So one convenient thing is you use things which are transparent. So you find a small fish from a rice paddy in the north of India, and you call it zebrafish, and it's transparent enough and you can see through, and you stuck GFP in it. You can clear it, and that's convenient. So you choose ways, so modifying your sample or putting more light in it, which is never a good idea, because in light sheet, you have always to deal with between this excitation decay and detection decay between the light sheet and the detections. So here are some examples. This is a stack, and you can see that if I look at the nucleus from the front and the nucleus from the back, going from front, back between the good side and the bad side of the illumination, you can see that on one side, it's good side of the detection. So you can see that both nuclei are good because I have more light coming into the systems. And then after, if you look from front to back, you can see that each time there is a decay between what you record. So if in that particular thing, so it's a single illuminations, you can see that I have a quarter of the image, which is pretty good for segmentation, but the back opposite side is not looking good, but I will still try to segment that. So remember, depending on the numbers of light sheets and how they penetrate your sample, you have a decay on excitation and detections. So, and this will affect many aspects, multi-view. The other thing you have to remember is how big is your sample. So if I, for example, in that zebrafish, have a zebrafish perfectly aligned, the size of my stack, numbers of plane I need, will be optimized. If my fish is like this or like this, then the size of a stack will be massive. So remember that the bigger your stack, the worse your nightmare will be. Because if you don't make an effort on trying to get as less plane as possible, you're going to increase those effects of illumination decay, detection decay, and the amount of data you're going to generate at the end, reaching the terabytes. And you don't want to spend your time moving data to the cloud or blocking everybody's email inbox because you're saturating the network, or carrying a hard drive up and down the stairs all the time for too many years. I've done that enough. Think before you record, not only in excitation decay, but also size of your sample. So here's an example showing you what happened when you use two light sheets. If you talk to manufacturers and all kind of people who design a microscope, oh no, having two light sheets is perfect, then you see everything. We use a sponge because sponges are absolutely terrible. They diffract like hell. And you can see that in the middle, there's a gap. If you use two light sheets coming from two sides, you cannot expect the light sheet to meet perfectly well in the middle. This is in your mind. This is a dream. This is a theory thing. As soon as you have two light sheets coming from two sides into your sample to optimize, things in the middle will not be as good as on the side because of excitation decay and detection decay. okay? So that's to show you, remember, there is a dodgy line in the middle, and that always happens. So don't send me an article saying, oh, things on the middle are different than on the side, because I would tell you this probably doesn't exist. Stop. Cut it in half, put it on the confocal, and you'll realize that all your nuclei are exactly the same size. And the light sheet just went the wrong way, and that's it. Stripes. Yes, everybody said, you know, each time I put my light sheet, things are being blocked. Yes, that's normal, because photons react normally. If there's something blocking them, they don't go through. It's not magic. And we're not talking neutrino here. We're talking photon. So stripes exist, but stripe exists also in confocal. But because you detect them into a very thick, thin stripes, you don't see them on the other side. So those stripes are also relevant because they tell you where nuclei are, where objects are, and so on and so forth. There is a way of dealing with it. So you can actually tilt the light sheet, you can scan. There's always ways to get around it. But those stripes are representative of the fact that the light sheet has to go through your sample, and there is detection and excitation decay going through. So now, geometry. As I was explaining, the SPIM is something like this. Now, an ultra microscope is like this. So the sample is flat. And then you can actually combine all kinds of different. So different light sheets will have different organizations of their optics, which means that your sample will never be at the same place. So when you talk about, for example, if I say a SPIM, I know that my sample will come from above like this, will be fixed, and then the light sheet will move through the sample, or the sample will move through the light sheet, one or the other. That's also very important, because if a light sheet goes through your sample is fixed and you don't have to move it. If a sample moves through the light sheet, it has to be fixed enough not to move, to shake. And many systems like the SPIM, the sample moves through the light sheet. So the importance of stabilizing your sample is much more important in a system like this than in a system which is scanning through the system. So be aware that depending on the system you use, there will be two possibilities. One, the light sheet go through your sample, and sometimes the sample will go through the light sheet because it's easier. So that makes a difference. Also, if your sample is on a slide, gravity will affect it. If it's suspended, gravity will also affect it, but in different ways. So depending on the geometry and the way you mount your sample, you will have different things. This is why you sometimes cannot compare between two light sheet systems, because one doesn't use the same mounting system or the same scanning technique. And geometry are absolutely mental, because you can now buy a 96 well plate with micro mirror at the bottom, so that you can create the light sheet with a confocal microscope using the plate in the 96 well plate. So you have micro mirror device, you have all kinds of light sheets. So it's why we have 150 acronym plus, is because you can create a light sheet with many aspects. You can use a prism, you can use 2 light sheets, 3 light sheets, 4 light sheets. So for example, the ultra microscope sold by Zenaya Biotech has 6 light sheets, 3 on each side. So what does that mean? So for on an ultra microscope, you are on an horizontal light sheet and your sample is being scanned like this. So your stack is like this. In a SPIM, your stack is like this. In a tilted, your stack is like this. In a single objective, your stack is like this, is skewed. So depending on your system, you will have also to change the way you process it. So in a single objective where you scan like this, you have to deskew it. So it has some effect on your data. So every light sheet scanned differently, generates slightly different format of stack, which have to be processed slightly differently. And somebody just ignore it, and that has some tremendous effect on some of the samples. So this is something you have to remember. Where is the light sheet? Where is the relationship between my light sheet and my sample, and how the stack is generated? So that's one more level you have to remember. So don't say, oh, I have a light sheet microscope. You have to explain, I have a light sheet microscope, which is a single objective, so I have to deskew my stack. And then people understand, oh, it's like this, and so on and so forth. So it means, depending on the way you have a light sheets, it will go through your sample differently, and you will have to mount your sample differently. So an ultra microscope is usually flat, but you could actually have a flat sample mounted in front of the light sheet systems by mounted on a pedestal. And for example, the Luxendo systems are mounted from underneath. On the SPIM, it's mounted from above, and you can have different things with agarose gel, Gelrite. You can even mount cover slips if you want. And on the tilted system, you can see that you can also do the same, as long as you remember what's the relationship between the light sheet and the objective and how the excitation decay and illumination decay will affect it. So as I reminded you before, the shape of your sample and the way you mount it will have some tremendous effect on the amount of data you record if you do single, multiple views. I should remind you that even though in light sheet you can do multi-view, less than 10% of the users need multi-view. They usually do a super duper multi-view for the cover of a magazine because they fail so badly at fusing, because of what I explained before in terms of how the sample are, that it doesn't work. And also because most part of the time you actually don't need it. And also multi-view means 4 to 6 times more data per time point. And that's make it absolutely ridiculous in terms of amount of data you have to manipulate afterwards. So one thing you have to remember before to go into multi-view and super duper thing is to calculate the amount of time you're going to spend on it. So sometimes you're going to record data, you know, over a week of a zebrafish. And then after you're going to spend 6 months trying to fuse it and get it together and publish it. So think twice before accumulating great data set and pretending, oh, I got a super duper experiment with 6 terabytes. Yeah, and after you have 6 months of pain trying to get that terabyte into one data point, okay? So having more data is like, you know, oh, my resolution is better than yours. That's usually useless. You know, we are scientists. If one view is enough, use one view. If you see what you see, if one view is enough. Multi-view just to show off and spending months trying to fuse it, because many of the biologist users are not image specialists and struggle with image processing, because that's not our training. That's probably not a good idea. So think twice before you mount your sample, optimize it to have as less plane as possible, as less data. The less data you have, the faster the data processing will be. And remember, we are in a situation of climate crisis. So using computer for longer, just because it's important for your data load, is probably not good for the planet. So think twice before throwing 64 terabytes on a cloud. Just a reminder about CO2. So you can use a single light sheet. So that's something which was done by Karakoglou. It's a very nice thing. It's basically they mounted a copepod, so it's a smaller shrimp, into a light sheet and let the copepod have several meals a day. And they basically let them into the fish tank for 48 hours and look how the copepod was able to digest. So as I explained before, you can have a single light sheet and see what happened through the light sheet over time. You don't need always to use the light sheets for scanning the volume. You can use single plane light sheet for one thing. So you could look at how the things change in shape. You could look at fluid dynamic around it. You could look at digestion process, movements of particle. So it's the way you use light sheet for doing FCS. So for fluorescent correlation spectroscopy, you can use dynamic, and you can use, in that case, digestion. You can also do depth. So you can do, in that case, we use the surface and the thing. So this is an aphid, so it's a small insect. So with a light sheet, we can see through, and you can see those vesicles in green, which is the way the new shed is going to come, and but also the surface. So with a light sheet, we can scan the surface and look inside at the same time. So you can actually use the plane and line to scan the object at once. And then you can actually, because you have it in 3D, you can actually turn it into a monster and 3D print it and have it in your desk. So this is an aphid that now is put in a database where people making 3D games can use it to make it a monster, but at least it's taxonomically correct. Because, you know, some of those insects into online games are pretty bad. So we are trying to use the light sheet to help them out with properly taxonomically correct animals. But it's also to show you that light sheet can be applied to taxonomy of small insect, nematodes, small crustaceans, and more people are using the light sheet as a line scanning object. So they actually scan the surface of the object. And we had a visitor from Australia on our last course, which was looking at a head of an aphid of a moth, I think, tiny moth, to be able to do the taxonomical analysis. So don't forget, light sheets also define a line, and the line can be used to scan surfaces. So you could also scan the surface of a spheroids if it's properly stained. So remember, the light sheet could be as a single plane, as a line for scanning, and then you can need as scanning. So there's different ways to mount, and I'm not going to spend a course on that because I usually spend 2 days on this. So the typical one is you put your things into gel, but some animals don't like to be in gel. You can suspend them if they are good enough. You can put them into a cuvette, and there's plenty of cuvette being developed by people like Francesco Pampaloni and so on and so forth. So there's different way of mounting. The problem is so far we don't have the slide. You know, now in most part of microscopy, everybody uses a slide, and you mount on the slide, and you put your cells on the slide and cover slip. Everything exists. In light sheet microscopy, we don't have suppliers providing us with a perfect mounting technique. So often in light sheet microscopy, you have to do a trial and error to get your sample right. Now there's more papers, and you can look into the nature of the methods in order to get that. But often you need to practice because there is no slides. So some of example you probably know from Jan Misken and others is using agarose, which you embed in agarose. Remember, the agarose is not for microscopy. The agarose is something which has been developed for DNA extractions. So, and what you usually do is you take the agarose from the shell, and the agarose is so bad because there's so much dust in it that you don't see through. So if you use agarose for microscopy, everything has to be sterile, under the hood, no particle, and make sure it's clean, because we got some very ugly thing. I can probably tell you that. So you have different ways of mounting, different in size. You have to remember that you need to have as less hydrogel as the sample, because the thicker the hydrogel, the same thing, there will be a detection decay as you go through the sample. Here is an example. This is showing you agarose at different concentration from different suppliers. So you can see that they come from different suppliers, but some are used for large bands of gel and so on, because agarose comes, there's about 80 different types of agarose you can buy, depending on what you want to do with it, because agarose is like pasta. There's long ones, short ones, big ones. So agarose doesn't come as one unit. So agarose and gel you use will be different. And I will show you now what happens if you take an agarose from a shelf that somebody has left for 20 years, and that's what you get. Because people have been putting their fingers in it, they have opened it, it's full of oxidations, and that thing goes really bad. So when you put something in a light sheet, especially when it comes from the side, it better be clean. So if you use gel and so on, make sure it's clean. Remember that if you put something in a gel, it's not going to like it. So, you know, there's restriction, compression, variation over time of your gel. So if you put a zebrafish into a gel, it's like, you know, you probably don't want to be in a pot of jam, you know, your head into it. It's probably not good for breathing. So think that the zebrafish is going through the same thing. So put yourself into the situation of a sample. You may not want to be stuck in a block of agar for 8 hours in a microscope. It is not very good. It has also some effect on diffusion. So if they excrete something, if they release something, it's going to stay around them, and that's going to cook them. You know, if they release, you know, excretions which are bad, that's also going to change the pH and so on and so forth. So if you put something in a gel or a cuvette, remember that, especially if you go over a long period of time, for weeks, for example, that's going to go bad. So remember, it's not because you see through that is good. Remember gravity. So if you go from above, things are going to swing. From underneath is going to swing like this. And if you go like that, it's going to go that. So depending on the way you mount your sample and the geometry of your light sheet microscope, things may go. We lost some sample in the things. The best example, we had a zebrafish swimming into the Z1 chamber for 2 days while we're doing a workshop, and you could see the shadows coming back from back and forth, which means that it was physiologically relevant, but not what we wanted. So you have different ways. So these tubes and FEP tubes and so on, which have been created. You can grow plants into a light sheet systems with light on top and things underneath. You can make your own cuvette. So that's a work from Francesco Pampalotti and Elizabeth Hertz, which you create your own cuvette at the size you want using FAP and basic instrumentations. So plenty of things are available, but you have to dig a bit. So you have 3D printed holders, which are available, that we are making, that we're going to post online in the coming months. You can do different kind of mounting. So that's another example of acoustic levitation. So they use sound to keep the fish above. I don't know which type of music, but I don't think I won't like to leave a zebrafish in AC/DC music forever. It's probably not good for his art. And I think I'm going to stop here because it's already quite a lot, I think, I've been talking about. And just one last thing, clearing. I just want to remind everybody, if your sample is not clear at the start, but clear at the end, it means something has happened. You have cleared it. So you have removed things. Make sure you know what you have removed. Because if you look, for example, and work on a perfect protein interacting with ceramide, but your clearing technique remove the ceramide during the process, what you're looking at is floating GFP. If you clear a sample, things happen to the sample. It may be convenient. It's like expansion microscopy. Expanding works. Did he actually keep what you wanted or not? Ask yourself. It's not me to choose, but in the clearing, remember what you lost in the process, because that could have consequences on your result. So clearing is a nice technique, but it does something to the sample. It's like, a dead sample, it's not the same thing than the live one. And clearing is the same thing. A cleared sample is not the same thing than the real original sample. I'll leave it here. Think about it if you use clearing in light sheet. We have a book coming up. We are nearly finished and it should be back at the end of the year. Normally we did that with Pavel Tomanchak. There's most part of people in the community going from imaging platform to others and so on and so forth. And I'm going to stay and stop here. Sorry, I've been too long, but I cannot stop. That's it. Thanks very much.
Thank you, Emmanuel. That was really, really excellent. Thank you so much for all these tips, information, and so on. As said, very excellent. And we have already the first question by Milena. Steve, can you unmute Milena? Or
Milena, go ahead.
Sorry, that was accidental. Sorry about that, Steve. Very nice talk though, Emmanuel.
Thanks.
Okay, let's see. Are there any other questions from the audience? Either raise your hand or write in the chat box or just speak out. Everything is welcome. I might start with one question, Emmanuel. You obviously explained a lot of challenges or things we have to consider. Where I would like maybe to know a bit more, if possible, also because I think it's one of the biggest challenges, at least I am aware of, you're facing, is with the data analysis. I mean, you mentioned already, yes, the amount of data you get will be really, facing a challenge for you. How are you in your lab or in your imaging facility dealing with that? If you can explain a little bit maybe what kind of PCs or setup you have.
So what we have is gaming PCs. So basically, because gaming PCs are cheap, I mean, they are fun because you get them with, blue fan and stuff, and everybody look at you. Those gaming PCs use GPUs, and now thanks to Robert Haas and the Fiji community, so ImageG, we have a lot of very nice piece of kit. So Big Data Viewer, for example, has been developed alongside Light Sheets by Tobias Pitch and Stefan Preibisch, and we use it. But in terms of setup, so first we have an optical cable between the microscopes and the processing stations. Because if we were sending 4 terabytes on the networks, nobody could send email for 8 hours. So, and usually when you buy a light sheet microscope, they usually give you an optical fiber for that reason. So, you can buy an optical fibers, and spend an afternoon running it through the ceiling, and it's far more efficient. than anything else. So yes, between the microscope and the system, we have optical fibers. We have a server, and we are brutal. So basically, when people record data on the microscope, they have to check them. They have to open them and do at least projection of their stacks. If it's not done, after 48 hours, it's deleted. We don't care. You don't keep data. If people don't care about the data, I should not care. And because of LightSheet, it's very, very big. So as soon as you record, you have to check them, because, you know, sometimes the zebrafish has disappeared after 2 days, you know, it's just fall and things. So why should you keep black pixel forever? So, and also it's a training exercise. Students, oh, but you know, if it's still the first few hours, I should keep it. No, you don't. If the experiment is not complete, is not correct, the data should not stay. Because if not, you're going to end up with hard drive piling up into your drawers on your desk with that black pixel, something you cannot analyze. So, and then after they have to move them to the server, and on the server, they have to be processed. If they are not processed for whatever reason, they also go. So if you have a project where you need, you know, 10 zebrafish over a certain amount of time, you have to, I mean, the groups, because we're not a facility, the group who has the data has to sort that out. It's why we tend to calculate the amount of data you're going to generate before you touch the microscope. Because if not, you have a user starting, you know, a series of 20 zebrafish over days, and then they plug everybody else, and then the PI come to you. Yeah, but it's very important. I don't care. It's important for you, but you cannot block everybody else. So with data, you really have to be brutal to make sure they record what they need to record. And there's ways around. So for example, now we tend to crop the data set. So everything around, because, you know, when you, for example, if you start a very embryo with 4 cells, it's going to take, you know, 100 pixels at the center, but the full frame is, you know, 1,000. So all those black pixels, you don't need them. So you can actually crop 10% around, and actually you only keep the data you need over time. And many people like, what's her name? Kate McDowell, which is looking at embryo development, she's actually cropping very early. If not, she cannot. So with data, be brutal and tell everybody why, and get an optical fiber.
Awesome. Thank you so much. It's really good advice. We have a few more questions. The first one, Juan, please speak out.
Hello? Can you hear me now?
Hello, Juan.
Hey. Great talk, thanks. Yeah, learned a lot. This is a bit of an aside, but the coral images were absolutely beautiful. And coincidentally, we are looking for an image to put on the front page of the Napari website. And I'm wondering if those are available somewhere. It goes very well with the Napari theme, right?
Yes. Oh, and I will be more than happy to support the Napari plugins. I've been playing with them. You can contact me, and I can arrange things, and we can go through the series of coral images we have, and I'm more than happy to support the Napari things.
Awesome.
So everyone, try Napari for LightSheet imaging. Visualization is a very nice thing to do. And it goes well with Fiji as well.
Yep.
Okay, awesome. Azel, I saw you had your hand up. It's now down. If you want to ask, just raise. Ah, there you go again. Awesome. Azel?
Hello, Emmanuel. Thank you so much. Excellent talk. My question's on terms of data analysis. So would you recommend when you're acquiring images to rather, I guess, not be generating lots of data so that it's easier to analyze in the long run? Yeah. So really thinking about your experiment prior to obtaining those large amounts of data.
Yeah, so the typical things we teach during the workshop is downsample, downsample, downsample. So the first thing is, once you get the data, before to start any kind of processing, you downsample, downsize your data set, you know, by a factor 2 or 4, and you try to process it. If you manage to process it, downsample, then you can go with big ones. Because if you start with the big ones, you know, everything bigger is a bit more difficult. So the first thing is think about the data you're going to generate, the numbers, and test any kind of image processing downsampled. Because what we have seen is people, oh, I've got 4 terabytes, I'm going to try. Yeah, sure, go on.
Yeah, that's
and it goes
exactly
wrong.
me.
So exactly. So you take your sample, you can downsample it. The quality will not be as good, but if it works, you know that it works. So you know that you can actually block everybody else access to the server overnight with your big data set the next morning, but at least you can send them a warning. You know,
Yeah.
guys, I'm sending the big thing. It's a long weekend. Nobody touch my server. So data set smaller as possible before you process them, just to make sure you put every good aspect on your side and not going blunt force.
Yeah. Yeah.
Blunt
Thank you
force
so much.
trauma is always bad in general in movies. So downsample, process. If it works, come back to the big ones and warn everybody that they cannot send email.
Thank you very much.
Thanks.
Awesome. Very good questions. Again, if there are any other questions, just please raise your hand and I will allow you to speak out immediately. I might have one more question, and maybe I didn't get it and so on, but you said at the beginning you might think of maybe that at one point there's a light sheet with 8 objectives, but then also you said they have to be in a 90-degree angle. So is it at all possible, or do you really think that will happen at one point, or was it more kind of a joke?
It's a kind of, I think it's a joke, but actually you could put 8 objectives only for the light sheets.
Ah, okay. Got it.
So for example, the system from Miltinia Biotech has 6 light sheets and 1 objective, so there are already 7.
Mm-hmm. Okay.
So somebody is gonna add 1. The problem is you do that for certain reasons. So the Miltinia Biotech, because they do clear large sample, they're trying to optimize the thickness of a light sheet uniformly around a large plane, is why they try to fit as many light sheets coming from different directions, like a circular light sheet, can creating a disk of light sheets. But you know, and also now with the single objective, you actually have back objectives. So you have a light sheet coming from a single objective, but it projects on another 2 sets of objectives. So for 1 light sheet, you already have 4 objectives.
Mm-hmm.
So if you actually do a dual single objective light sheet, you end up with 8 already, but at the back. You don't see them on the system, but they are at the back on the back focal plane illumination systems. So it's, unfortunately, somebody's going to come out with that. You know, they cannot resist. It's so tempting, you know. Just want to point out a couple of things. First, there's the blog with Elizabeth. And currently, on my Twitter account, I'm running a 1 pane a day, 1 acronym a day. So I'm going through all the acronym from light sheets from the origin to now. So we are in 2012 and it's getting mental. So if you want to have fun every day with an acronym and trying to guess it, during the workshop, we have a game show, which is guess the acronym. And it's absolutely hilarious because nobody can make sense of the acronym anyway. So it's just, so if you want to have a bit of fun and understand how light sheet works, I'm running on my Twitter account 1 acronym a day, which help out going through the difference, and then you're learning about different type of light sheet and how they work. Unfortunately for this year, the workshop is complete, but in 2 years, we'll have another one. And there is now one in the US, run by Gary Levsky in MBL and Woodall. And we're hoping to expand a workshop in South America, and hopefully maybe we will be able to get one in Australia at some point if we swim well enough and if the currency now in favors.
Yes. So what's your Twitter name?
It's Reynaud Emmanuel.
Okay.
So it's quite easy to guess.
Yeah. Okay. Awesome. All good information. So it seems there are no further questions. So Emmanuel, thank you so much for really an excellent talk where everyone learned really heaps and heaps. We're sure also as it's recorded, it can be re-listened again if anyone wants to hear it again when they start maybe imaging some samples, to listen again first to this talk before starting imaging. So thanks again. It was a real pleasure having you here, and yeah, hopefully see you soon, maybe at a workshop.
Yeah, it's perfect now. The tide is good, so I can go swimming before everybody wake up.
Absolutely.
Thanks for having me. Enjoy the evening, and if you need to reach me, you can find me on Twitter, and also Jennifer has my account, and I will send her the link to the blog. So if you want more information on light sheets, please don't hesitate. Thank you very much. Have a lovely evening, and cheers.
Thank you.
Thank you.